In a lab outside Cambridge, scientists took skin cells from a 53-year-old woman and made them behave like they belonged to a 23-year-old. They did not use a cream or lasers. This was done with a switch, flipped at the genetic level. Today, I'm going to show you exactly how they did it, what it actually means for you, and why some scientists are calling it one of the biggest breakthroughs in the history of aging research. It's 2022. At the Babraham Institute, a research campus near Cambridge in the UK, a scientist named Dr. Diljit Gill is staring at data he's been collecting for years. First as a PhD student, working in the lab of a well-known epigeneticist, Wolf Reich. Gill's question was deceptively simple. Could you make an old cell young again without turning it into something else entirely? Because here's the problem scientists had run into before. You could reprogram adult cells all the way back to stem cells. Cells so young, they're basically blank slates, capable of becoming almost anything. But when you do that, the cell forgets what it was. A skin cell reprogrammed this way stops being a skin cell. It loses its identity. Gill and his team wanted something different. They wanted to rewind the clock without erasing the cell's memory of its job. The technique is called partial cellular reprogramming. Instead of pushing the cells all the way back to a stem cell state, the team applied the reprogramming process in a carefully controlled, shortened way, enough to erase the marks of aging, but not enough to erase what the cell was. The results were published in the peer-reviewed journal eLife in April 2022 and confirmed independently in reporting by Live Science, ScienceAlert, and the Babraham Institute itself. To prove this wasn't just a visual trick, the team measured age using two hard, quantifiable tools. First, the epigenetic clock, which is chemical tags on DNA that reliably indicate a cell's biological age, almost like rings on a tree. Second, the transcriptome, essentially, the full readout of which genes a cell is actively using. By both measurements, the treated skin cells matched the profile of cells roughly 30 years younger than their starting point. That's finding number one. This isn't anti-aging marketing language. Scientifically measured, this is age reversal at the cellular level. This is the most interesting and honestly more useful. Looking young on paper means nothing if the cells don't actually work young. So, the team tested function. Skin cells called fibroblasts have one especially important job: producing collagen, the protein that gives skin its structure and helps wounds heal. The rejuvenated cells didn't just match young cells, they outproduced the untouched control cells. And in early wound healing tests, these reprogrammed cells moved into simulated wounds faster than older, unreprogrammed cells. Dr. Gill summarized it plainly. Cells can be rejuvenated without losing their function, and rejuvenation appears to restore that function to old cells. And notably, researchers also observed a reversal in aging-related markers connected to genes involved in certain diseases. That detail matters. It's why this research isn't being filed under skincare. It's being filed under regenerative medicine. So, who does this matter to, beyond scientists in white coats? A burn victim, years from now, whose damaged skin cells could be regenerated using a version of this technique, healing tissue that today simply can't repair itself well. Someone with a chronic, hard-to-heal wound, a real and serious problem for people with diabetes, for example, where slow wound healing can lead to severe complications. And more broadly, picture the millions of people affected by age-related diseases. Because this technique didn't just make cells look younger. It reversed markers tied to genes associated with age-related conditions. That opens a door toward therapies for conditions like arthritis, And researchers are already cautiously exploring whether similar reprogramming approaches could someday apply to neurons, the cells in our brains tied to memory and cognitive decline. To be clear, none of this is available in a clinic today. This is early-stage foundational research. But the scientists behind it were careful to say something important. This is not a magic cream, and it's not immortality. It's proof of a mechanism. And in science, proving the mechanism is usually the hardest part. Let's go back to that 53-year-old woman whose cells started this whole conversation. She'll never know her exact role in this. Her donated cells were anonymized like all research samples. But somewhere in a freezer in Cambridge, a tiny sample of her skin behaves on a molecular level, 3 decades younger than the rest of her. That's the strange, almost poetic part of this story. Aging for a long time felt like a one-way street, something you could slow down maybe, but never actually reverse. This research is one of the first hard, peer-reviewed pieces of evidence that the street might run both ways. Not for a full human body, not yet, but for a dish of living human cells, this happened. So to recap, scientists reversed 30 years of biological aging in human skin cells, confirmed it 2 different ways, showed the cells still worked better than before, and it could one day help everyone from burn patients to people fighting age-related disease. Is this the fountain of youth? No. Is it one of the most important aging discoveries of the decade? A lot of scientists think so. If you want to see how this same technology might apply to the brain, memory, and neurons next, hit subscribe and drop a comment. Would you try cellular rejuvenation therapy if it existed today? Let's talk about it below.